Hello, everybody. My name is Justine Smith.
And I'm Kelsey Hopkins, and today we're going to be talking about the effect of wild pig removal on natural resources in the southeastern United States. Our co-authors are Dr. Michael Minjak, Kim Sash, and Dr. Mike Connor.
To begin this presentation, I wanted to provide you with a brief background on our target species, which is the wild pig. It is sometimes commonly referred to as the feral swine or feral hog, but throughout this presentation, we're going to call it the wild pig. It is a introduced worldwide invasive species that is originally native to Eurasia and North Africa. It was introduced to the United States in the 1500s by Spanish settlers for agricultural use. Unfortunately, escape from pens and illegal translocation facilitated the expansion of the wildlife population. As of 2019, wild pigs have been reported in 35 states. To further exemplify their population expansion, in 1982, wild pig populations were estimated to be at 2.4 million, and in 2016, they're now estimated to be at 6.9 million. Wild pigs are a very successful invasive species due to their high reproductive rates, their early reproductive age, their low mortality due to lack of natural predation, and their opportunistic and generalist diet. This generalist diet can harm native flora and fauna. Fauna as an example of white-tailed deer or other game species as they compete for resources like acorns or overtake feeders for hunting. Wild pigs are also a threat to the agricultural industry, and as you can see here, annually they do about $150 million worth of damage in Georgia and $190 million of damage in Florida. They also pose a huge human health and wildlife health and safety risk through potential transmission of diseases and parasites through indirect and direct contact. As an example, wild pigs can facilitate the spread of E. coli when in contact with water sources. When levels are high enough, it becomes hazardous to any animal or individual that utilizes that water body. Damages to industries and the threats to native flora and fauna are carried out through three primary damaging behaviors. The first behavior that is exhibited is rooting, as you can see in this video below. Pigs will forage for food by uprooting soil with their spade or triangular-shaped snout. This can also be referred to as grubbing. Some studies have shown that rooting facilitates the expansion of invasive plant species, a decrease in native plant diversity, and alters nutrient availability for plants in the soil.
And to continue on with what Justine was saying, another type of damage behavior that is commonly found in areas with high pig populations would be rubbing. When pigs find a sturdy, upright object such as large trees or telephone poles, they will rub their bodies up against it for a couple of reasons. They can do this to remove ectoparasites, to mark territories with the scent glands on the top of their heads, or to simply scratch an itch that can't be reached. You will more than likely find rubs at the base of trees where wallows have been formed. So wallows are your typical mud hole, give or take some pig tracks or possibly rooting nearby. Pigs will use these mud holes to decrease their body temperature and, like rubbing, keep parasites off. There's not a lot of research done about rubbing specifically, but some studies have shown that rubbing and wallowing can impact tree health, tree quality, and overall tree growth development. Now I'd like to talk a little bit about the initial start of this project. A little background for you. In 2014, the US government granted the USDA's APHIS program a multi-million-dollar grant to begin managing for wild pigs in the United States. Jump to 2018, the Feral Swine Eradication and Control Pilot Program, or FSCP, is now authorized by the Farm Bill, a well-known bill created to provide cost-effective management strategies for landowners. Now skip to 2020 and current times, the USDA now oversees a multitude of projects across the United States. Today, we're going to be talking about two individual pilot projects overseen by Tall Timbers Research Station, the Jones Center at Itchway, in collaboration with UGA's Warnell School of Forestry and Natural Resources and the Flint River Soil and Water Conservation District.
Thank you for that background, Kelsey. So to kind of segue into our projects and our objectives, I will be going first. And I would like to say that USDA will be responsible for removal events on our properties throughout the duration of both of our projects. Removal events primarily consist of aerial shooting events, along with consistent trapping and night shooting. So the objectives of my part of this project can be detailed in these 4 points here, where we would like to document wild pig response to these removal events. We want to assess the response of wild pig removal on selected native vertebrates. We want to quantify agricultural crop damage, and we also want to assess changes in native vegetation in response to these removal efforts. So my study area is in the Albany area of Georgia, which is located in Calhoun County in southwestern Georgia. I have 4 properties total involved in my project, ranging from 1,500 to 7,000 acres, totaling 30,000 acres of land. Landscape types that are primarily seen throughout my properties include longleaf pine and loblolly pine stands, mixed hardwood, wildlife openings like food plots, riparian areas, and scattered wetlands. These properties were selected based on knowledge of pig abundance in the area collected by the USDA Wildlife Services. 3 of these properties support and encourage hunting during their appropriate seasons. All of these properties have undergone some form of historical landscape management practices, some of which including prescribed burns, installation and utilization of wildlife food plots, and timber harvests. In addition, all of these areas have experienced some form of negative impact from wild pig populations and have used a variety of management techniques to control or attempt to exterminate these populations from these locations. So, how do I want to fulfill these objectives? My study is a passive camera trapping survey, which means that these cameras are unbaited. I currently have 147 cameras spread across my 4 properties, and they have been in the field since June of 2020. Camera locations are based off of a 100-acre grid system overlay created in GIS. In the field, I identified wildlife trails within each of these grids that corresponded with pig signs, rooting, rubbing, or wallows. And then from there, I would find an appropriate location to place my camera. I am currently using Browning HD Strikeforce Pro cameras set with a 30-second delay and are motion triggered. At these camera locations, I am conducting vegetation sampling and quantifying ground cover percentage using a 1 meter squared quadrat. This takes place in every cardinal direction around 10 meters away from the camera location. In these locations, I am documenting presence or absence of wild pig damage and using a Robel pole method to record the average and tallest plant heights in that location. Vegetation sampling takes place 6 months apart during the growing and dormant season, or during June and January, roughly. For agricultural field sampling, I will be sampling 1 field per crop per property, totaling 4 fields. And I will be using drones to conduct this sampling. I have 4 proposed sampling methods to carry out. The first method is through flying 3 randomly placed transects in these designated fields. These transect locations are generated through the use of GIS. The second method involves randomly generated waypoints and taking pictures of these randomly generated waypoints through GIS in these fields. The third method is ground measurements that also takes place at these randomly selected waypoints. On the ground, I will be documenting presence or absence of pig damage, measuring average vegetation height, recording presence of species through tracks if available, and determining the source of damage, either from wild pigs or other non-target species. And finally, the fourth method in which I will be implementing is flying the drone around the entire perimeter of the field and having the camera facing inward to try to view the field in its entirety. These drones will be flown on a monthly basis after the fields have been planted. Finally, all of the images that are collected for my study of all animals and individuals are documented via an internal system called Oracle that is provided to me through the Jones Center at Itchway.
Thank you, Justine. The overall objectives for the portion of the project overseen by Tall Timbers are to develop a successful pig reduction program in the Red Hills region, to reduce pig populations by at least 75%, to reduce pig damage by at least 80%, and to monitor water quality improvements throughout the duration of the project. These objectives are expected to be met by 2023. So my portion of this project covers the Red Hills region of northern Florida and southwestern Georgia. This area supports diverse management practices for agriculture, forestry, and large recreational hunting properties. The initial project area was split between focal areas or areas with high pig populations and landowner participation areas. The map shown here indicates both of those. The USDA has provided trappers that are actively trapping pigs on all of these properties, moving from east to west. While trappers are on the ground, I am monitoring responses on the 9 colored properties. 6 are in Leon County, Florida, and Jefferson County, Florida, and 3 are in Thomas County, Georgia. Tall Timbers is located in Leon County, Florida, and shown under the red arrow for reference. Each property is upland pine dominated with various ag fields, fallow fields, planted longleaf, and wetlands. Although individual properties are managed differently, some common trends have been prescribed fire, selective timber harvest, and previous hunting pressures on wild game and pigs. Similar to what Justine has going on, I also have a passive camera survey. Currently, I have 151 cameras deployed across the 9 properties shown previously. They were all initially deployed between October and November of 2020. The cameras are Moultrie M50 brand with a 1-minute delay, low sensitivity, 3 triggered multi-shot setting. Camera locations were predetermined using randomly generated points through GIS, and they are checked on a monthly schedule. Checking cameras can include, or do include, swapping out SD cards, documenting battery life, and clearing any new vegetation that could potentially trigger the camera. Cameras are used to determine wild pig populations pre, during, and post-trapping efforts. For every camera set, there are 4 150-meter long transects radiating from the camera in all cardinal directions. Every month while cameras are being checked, these transects are being walked. For any wild pig sign that is found, the habitat, GPS location, and type of damage is documented. Water samples are also collected monthly from designated wetlands while cameras are being checked. 2 bottles are filled with sample water. 1 is for E. coli analysis and 1 is for pig environmental DNA. After water is collected, 1 bottle will be plated on a Petrifilm plate and incubated for 24 hours for E. coli analysis. The second bottle is stored in the refrigerator for later transport to the Veterinary Diagnostic Lab in Tifton, Georgia. There, the diagnostic lab will extract the eDNA samples and report back to Tall Timbers with results. Samples are taken to Tifton once a month, while E. coli sample reading is continuous as we continue to monitor on these properties. All images are filtered and processed through Camelot imagery software. Each animal, excluding non-game species, are documented and counted.
And now we have some slightly exciting news to share of the current results that we can offer to you. So as part of my project, for the last 8 months, we have collected about 205,000 images. This total imagery also includes just photos of vegetation or of the sky when your camera gets knocked down. When filtering out these little mishaps, I have about 150,000 images containing animals. Now, currently, we only have a little bit over 1 month of images entered into our database. And from those images, we were able to find that a total of 19,000 animals were found in 1 month's worth of data. And from that amount, we were able to see that we saw 19,000 pigs.
I'm not too far behind you, are I? Tall Timbers has currently collected 116,800 images, including blanks and vegetation. Out of that 116,000, almost 27,000 are images of animals. And of that 27,000, a little over 1,500 are wild pigs. We have collected a total of 43 water samples. Of those samples, only 1 has tested positive for wild pig eDNA. And the highest amount of E. coli that has been counted has been 70 colonies.
Near the end, for everybody involved, we would first like to extend great thanks to all of the entities that have helped create these projects. So we would like to formally thank Tall Timbers, the Jones Center at Itchaway, the Flint River District, USDA, and the University of Georgia for providing us with this opportunity to conduct research and to further our own education. On my side of the projects, I would like to really extend thanks to my first summer intern, Samantha Nunn, of last year of 2020. She helped me navigate the fields for the very first time, work out my methods, and really get the ball rolling in a sense on my project. To continue, I really want to thank everybody involved at the Jones Center's Wildlife Lab, and all of the members included, specifically Gail Morris, Marlena Hamilton, and Kurt Siegler, who assisted me and checked my cameras and provided me with guidance and information while I was attending school in the fall. I also want to thank my current technicians, both Sarah Drake and Bianca Softoy, who have continued to provide me with assistance, with feedback, and check my cameras while I was able to attend classes this spring. And I would further like to thank those members and everybody involved in the Flint River District, but specifically Perry Cooper and Marty McClendon. These individuals have really helped start up this project and have really helped me communicate with landowners, find my properties, and helped me coordinate everything. So thank you so much.
I would also like to thank a couple of people. First would be Seth Williams for taking time out of his busy schedule to help me get my cameras on the ground during the start of this project. I would further like to thank the rest of the land management crew at Tall Timbers, Eric Stoller and Andrew Chase, for letting me steal Seth initially, and for helping me out with any of my struggles with any tools or any machines that I've needed during this project. I would also like to thank my tiny but mighty crew of interns, Mackenzie Hallmark, Alexa Murray, and Deanna Rienzi, for all their hard work, their constant positivity, and their perseverance.
And to kind of finish things off, if anybody has any questions, please feel free to email one of us or both of us. We're very excited to do this presentation and to answer any questions that you may have. However, just as a funny aside, we have two questions that we know we don't have the answers to. And one of them is, how do we constantly keep getting stuck in mud? To which it still baffles me today. And then the follow-up question, answer we don't know is how to properly get out of it, as I always need assistance to get out of it. And that's it.
We're good.